Hello everyone again, and thank you very much for the invitation, Katerina and Nikos and Sofia Schiza. And I'm very happy to be here among good friends and in Crete. And over the next minutes, we're going to discuss about biologics in COPD, the pro part of the discussion. So, as we discussed today, COPD is a very burdensome disease. It is associated with adverse outcomes. We do have a lot of treatments. We don't have the final answer yet because patients still have exacerbations and still have symptoms and still have disease burdens. And I believe that one of the main reasons that our treatments remain suboptimal is the complexity and heterogeneity of COPD that necessitate personalized treatment approaches. And for this reason, there is an urgent need for new treatments and new personalized targeted treatments, and I think the biologics can give us that. Here are my conflicts of interest. So we know that about 20 to 40% of patients with COPD have an eosinophilic phenotype, meaning that they have T2 inflammation. And as we discussed earlier today, inhaled steroids can improve the outcomes of these patients, and treatment response of these patients is associated with a reduction in blood eosinophils. So it seems that it is their action on this type 2 inflammation that helps people with COPD. So it is only logical to explore the safety and efficacy of biological treatments. And as we can see here in this slide, a lot of trials are ongoing and some have already been completed, phase 3 trials, looking at the interleukin-5 pathway, the interleukin-3 and 14 pathway, 4 and 13 pathway, and the alarmins. And I will focus my discussion on the results that we have from the big phase 3 trials around the interleukin-5 and 4/13. And I think what I will try to do is to resolve or explain this controversy in the results of these trials that we all have heard about by now. I'll start by the trials, the two main trials supporting dupilumab or Dupixent. The two trials are Boreas, that was published last year, and Notus, that was published this year, and each have around 935 participants. And in order to understand the differences in the results of the various trials, I'll focus a bit more on the inclusion criteria because that's, I believe, the key. So for dupilumab, these are the latest of the groups of trials around biologics for COPD that have been published already. We have very focused inclusion criteria of patients with clear type 2 inflammation and with clear burden of disease. So we have moderate or severe airflow limitation. We have MMRC of at least 2. We have high eosinophils above 300, equal or above to 300. And we have at least 2 moderate or at least 1 severe exacerbation. And we have a history of chronic bronchitis for 3 months during the preceding year. And importantly, we also have patients that were already optimally treated with the available treatment, so triple combination of inhalers. And we have people with current asthma excluded, and we have the smokers capped at 30%. And that's because current smokers are supposed to be more resistant to steroids, so perhaps there was concern about resistance to biologics as well. And we can see very positive results here, so both in Boreas and in the Notus trials, we can see that the risk of moderate or severe exacerbation was clearly reduced with dupilumab by 30%, so the risk ratio was 0.7 for Boreas and 0.66 for Notus, with confidence intervals that do not include 1. So important positive findings. We can also see a significant improvement in the pulmonary function test with improved FEV1 by 80 mL in both studies. And we can see improvement in the health status as well, with improved St. George Respiratory Questionnaire by -3.4. That's not above the minimum clinical important difference. And when we look at the responders analysis, actually, we do find positive results for Boreas, but not positive for Notus. So 51.5% of patients responded in Boreas compared to 43.2 in the placebo. And the percentages here were 51.4 versus 46.5 in the placebo. Importantly, for safety, we don't have any safety signals. The serious adverse events were similarly distributed between dupilumab and placebo. Actually, we can see that in placebo we have slightly higher rates of serious adverse events, and no other serious specific adverse event was highlighted. And in addition to the data that we have for COPD, obviously we can inform our knowledge from data from asthma, that we have more data about all biologics, and I'm not going to repeat that again and again. And we know now that biologics are safe choices of medications. I wanted to focus on two important subgroup analyses, and the first one was around FeNO. So dupilumab focuses on interleukin-4/13, so the alternative pathway. So it is anticipated that it would be more effective in people with higher FeNO. And indeed, the investigators looked at that in both Boreas and Notus, and it seemed that the treatment effect was focused on, was based mainly on people with higher FeNO and not lower. And then the second subgroup analysis I wanted to show was around smoking. And while there was a bit of concern there because the investigators have capped the participation, we can see that there was no significant between group effect. And now actually dupilumab with 2 positive studies is applying for FDA approval. Then we go to benralizumab with 2 very large studies, Galathea Terra Nova, that included huge populations, unselected even for eosinophils, and we see that they only had a subgroup of people that were required to have blood eosinophils above 220 and tested 2 or 3 doses of benralizumab. And we can see here that the inclusion criteria were a bit broader, so a bit less breathlessness. We remember that the MMRC was 2 for dupilumab, it's now 1. Again, history of exacerbations, which is important. But the other thing is that they accepted people that were not already on inhaled steroids, so not optimally treated previously. And with the exception of a borderline significance of benralizumab in reducing moderate or severe exacerbations, this trial was impressive. These trials were impressively negative. So no other signal in the exacerbations, no signal in the pulmonary function, no signal in the health status, and the good, obviously, finding was that there was no safety signal as well. And here, I think it's the problem. And if we want to be fair to the investigators of the benralizumab trials, we have to say that they did their work before the dupilumab study. So they lead the way in reality. They studied a broader population, and then in this very nice post hoc analysis, they started looking at what types of patients seem to respond. And if we observe these subgroups, we can see that people with eosinophils and previous exacerbations have some protection against exacerbations. If we add the triple treatment, so already optimized treatment, we have a bigger signal. If we look at people with a lot of exacerbation, then we can see a clear signal. So obviously, that's post hoc findings, not as strong as the dupilumab studies, but it seems that the signals are very similar in the big picture. And now we have the Resolute trial, which is ongoing for benralizumab, trying to establish the positive effects in the more selected population. And you will see I'm going backwards in the timeline, going back to the first studies, mepolizumab and Nucala. Metrio and metric studies, a bit smaller in the overall population. Metrics also had non-selected eosinophilic and non-eosinophilic, while Metrio only had eosinophilic patients. And we can see that their inclusion criteria were, well, probably in between, because they did have the exacerbation history as an inclusion criteria. They did require background ICS use and at least 2 other classes of COPD medications, and excluded asthma, obviously. So, we had an interesting observation here before I present the main outcomes. In the METRIX trial that was more unselected, we can see that more people in the mepolizumab arm discontinued treatment during the trial, 19 versus 13%. But this trend is exactly the opposite in METREO, where the population was more focused. It was only the eosinophilic patients. And we can see here that in the overall population of the METRIX study, so this includes eosinophilic and non-eosinophilic, there was no significant difference. When we look in the eosinophilic phenotype in both trials, METRIX and METREO, we can see that they reduce the incidence of moderate or severe exacerbation, same as the other trials. And we can see from the secondary outcomes that METRIX showed a trend toward reduced time to first moderate or severe exacerbations in the eosinophilic population, and the other outcomes were negative, mainly because of they were negative. And we can see again, similar to the benralizumab studies, the authors did another interesting post-hoc analysis looking at the interplay between the eosinophilic phenotype and treatment response, and we have seen exactly the same graphs for inhaled steroids, and we can see that the higher the blood eosinophil, the more treatment effect seems to have in patients with mepolizumab. Again, no safety signals. And again, because the initial trials were negative, now we have another ongoing phase 3 trials looking at mepolizumab. Before going to my take-home messages and trying to summarize all this, I just want to highlight that for asthma, we do have at least in the UK, we have managed to pull together really good MDT teams to try and identify those patients that are that would respond to biologics. And we have teams of nurses, physiotherapists, pharmacists that assess a lot of different aspects of the disease. And I think that we are missing from COPD, and that's the big next question, to see that the patients have already been taking their treatments correctly, to see whether there are other aspects that affect their exacerbations or symptoms. And I think in patients with COPD, the main thing will be the conditioning and sarcopenia or breathing pattern disorder. And the other thing that I kept for last, because it's not very good for me to say, it's good for Dr. Hillas actually, is that none of the trials really included patients with severe comorbidities. So patients with COPD do have severe comorbidities. That's an area that we need to assess, especially for safety. And going to the take-home messages, biologic treatments appear to be safe and effective for carefully selected patients with COPD. We know now that dupilumab can reduce the frequency of exacerbations, improve lung function and symptoms in patients with COPD, with high blood eosinophils, a history of exacerbations despite receiving triple combination. And we have in the back of my mind that it's important to assess FeNO as well, and the people with high FeNO seem to be the responders. For mepolizumab and benralizumab, they're slightly behind, mainly because they started earlier and their designs were not as optimal, but I think that their route ahead is clear, and again, we need clearly selected population to have a very, very good effect. Thank you.
Dear professors, dear friends, I am very happy to be here among all of you. Thank you very much for this kind invitation and for the opportunity to talk about biological COPD with my friend Alexander. I don't disagree with him, but have to remember to all of you that pros and cons derived from the Latin words pro, for, and contra, con, against. So I will support the against argument regarding the use of biologics in COPD. As we all know, COPD is a heterogeneous complex disease as a consequence of complex host environment, allergen, pollution, pathogens, but mainly smoking, interaction across spatial scales within the host over time. So typically, increased CD8 T cells and neutrophils are seen within the airways of patients with COPD. But there is an amount, a significant amount, 10 to 40%, with COPD patients demonstrate increased eosinophilic inflammation, allergic and non-allergic T2-mediated COPD inflammation. This is the cohort from the ECLIPSE study, where 37 of COPD patients have blood eosinophil count persistently over 2%. In any way, patients with COPD and eosinophilic inflammation are a minority. And this is from the last issue from ERJ, where Polverino and Sin proposed a pathobiology of type 2 inflammation with a cascade of events linked to lung pathologies, such as mucus metaplasia, small airway modeling, and alveolar cell apoptosis. In this way, in this cascade, there are several potential therapeutic targets: ADI-IL-33, ADST2, TSLP, IL-4, 5 receptors, etc. Okay, and the story begins with two negative studies. About 17 years ago, Professor Kostikas showed this data before. In this study, subjects with moderate to severe COPD received infliximab or placebo with primary endpoint the CRQ total score at week 24, as we can see from the figure, did not benefit from treatment with infliximab COPD patients. Nevertheless, this study stopped early because, although not significantly statistically significant, more cases of cancer and pneumonia were observed in the treatment with infliximab. And 10 years later, we have this study with an IL-70A monoclonal antibody with primary efficacy point that changed from baseline in pre-bronchodilator percent predicted FEV1 at week 16. As we can see, we have another negative study. And then the story continues, targeting. Targeting firstly, type 2 inflammation. And we had, Alex showed this data, MITREX and METREO study, mepolizumab or placebo, with primary endpoint the annual rate of moderate or severe exacerbations. Mepolizumab was associated with a lower annual rate of moderate or severe exacerbations, but actually we have METREX, a positive study, only patients with blood eosinophils at least 300 per cubic milliliter. And we have METREO, a borderline positive study, greater effect in patients with blood eosinophils at least 300 per cubic milliliter. And this efficacy signal was noted only for events treated with oral corticosteroids. So FDA did not approve mepolizumab, for COPD treatment. Then we have another negative study regarding benralizumab, Calathea and Terranova. Again, the primary endpoint was the annualized COPD exacerbation rate ratio, a totally negative study. And we have, of course, dupilumab, the only biologic agent with positive results for treatment of stable COPD. Borea study, primary endpoint, again, the annualized rate. Yes, indeed, dupilumab resulted in a lower annualized rate of exacerbations and better lung function than placebo in COPD patients with stage 2 inflammation. And the next study, the notes had the same results. And the story comes to an end when biologics targeting neutrophilic airway inflammation in patients with COPD. First, we have itepekimab, an ADI-IL-33 agent, in current or former smokers, with primary endpoint, again, the exacerbation rate. And as we can see, only in the subgroup of former smokers, itepekimab reduced exacerbation rate and improve lung function. The primary, however, endpoint in the overall population was not met. Astegolimab, an anti-ACE2 receptor of epithelial-derived IL-33, again with primary endpoint the exacerbation rate, and we have again a negative study for astegolimab. And finally, we have tezepelumab. In the overall population did not reduce the annualized rate of moderate or severe COPD exacerbations, only in a subgroup of patients with blood eosinophils at least 150 cells per microliter in this post-hoc analysis showed some greater reductions in exacerbation rate. And we have ongoing trials, but as we can see for IL-5, results non-significant, IL-5 receptor non-significant, and unavailable data about TSLP and IL-33, epithelial other means. And if we see the timeline of medication entering the market for COPD post-2020, there is a big question mark. And if I see good recommendations about the treatment of stable COPD, initial pharmacological or follow-up pharmacological treatment, no biologic agent is included in the algorithms, only LAMA, LABA, ICS, samaroflumilast, and azithromycin. No biologic agent is included in the treatments with evidence of reduction in mortality. To conclude, COPD patients with T2 high inflammation are a minority. Biologic agents for T2 high COPD failed. Only dupilumab had positive results. Biologic agents for T2 low COPD also failed. All these studies had as primary endpoint the exacerbation rate. As we all know, COPD exacerbations are heterogeneous events that may normally be easy to target. And finally, biologic agents are expected